And up to date, we have 70 talks. So it's really exciting time. I'm really glad that we get to have this meeting, even though we can't meet with each other. And I hope everyone staying safe as well. So today we have a very important and also very well-known scientist in the extracellular vesicles field, Michael Pffafl. So thank you for being here. He is a professor of molecular physiology at the Technical University of Munich, School of Life Science in Freising, Weihenstephan, Germany. Today, he has reached the principal investigator status at the Institute of Animal Physiology and Immunology, and is one of the leading scientists in the field of molecular physiology. His research focus is diverse, including gene quantification, RT-qPCR technology, RNA sequencing, extracellular vesicles biology, of course, and complex data analysis by integrative biostatistical methods and multivariate algorithms. So today, he's going to give us a lecture on EV-associated microRNA biomarker signatures in liquid biopsies, from the discovery, validation, to the future clinical application. So let's welcome Michael. Thank you.
So thanks, Carolina, and thanks, Jan, for inviting me to this WebEVTalk. It's a real pleasure to present our results, what we gained in research throughout the last years. And I was asked to tell you a story, the story about the development pipeline for microRNA biomarker signatures. And so I will introduce you through the last few years where we started this journey in 2015 up to today. And we had a lot of publications on the way. We produced a lot of publications, but we had a few questions, and we answered this question by these publications. So the first question was very early 2017, our cellular and extracellular microRNA, are they blood compartment specific in diagnosis? And our research topic was during these days, and still now, it's pneumonia and sepsis and connected diseases. The second question was how to optimize the methods to get the best answer. So the evaluation of serum, EV, isolation methods. The next question was, is there a difference between arterial and venous sera? The next question we had, how to standardize the entire workflow to make it more reproducible and of course more reliable, since in the age of liquid biopsy, a lot of methods are published and we want to standardize it in the best way. And the last was then the final, the kind of physiological paper we've done during these years. This was the outcome, the diagnostic potentials of circulating free microRNAs for community-acquired pneumonia and pneumonia-related sepsis. And as well, we had a lot of publications which we wrote with our groups and group members, mainly about QC and methods, which I will mention just shortly, like the MySIF guidelines, the functional analysis of EV, vesicle RNA, EV track. Of course, a validation, it's always a matter in our group, so the MICI compliant validation of the signature, and as well review papers from workshops and how to isolate EVs, and as well which goals and pitfalls EV vesicles may have. So let's start the journey at the very beginning. Of course, liquid biopsies in the center, the circulating biomarkers, and then we focused on circulating nucleic acids, and at the end, extracellular RNA. Everybody knows this slide. In liquid biopsies, DNA, microRNA, exosomes, or circulating tumor cells are important, and we focused on the microRNA in the beginning. Today, we are also focusing on mRNA expression, and we're also gaining for long non-coding RNA, so to create a kind of transcriptional network in these, not exosomes today, we call them EVs, in this interesting population of small EVs. of course, for diagnostic purposes and later prognosis. This is more or less our main goal of these studies. So why are EV associated microRNA? And we want to detect a biomarker signature, a molecular biomarker signature, which can measure a physiological or pathological change and use it as an indicator. These patterns should be directly regulated to a certain disease or also do a disease staging. And these molecular signatures should be precise, reproducible, and should be allow a fast evaluation in an objective manner. What are the advantages of microRNAs in comparison to other nucleic acids? Okay, the biopsy is non-invasive. It's mainly taken from liquid biopsies. We are analyzing short molecules. Short molecules means that are stable molecules. If you compare that to long non-coding and mRNA, they are kind of 100-fold shorter, and hence they are much more stable and also protected in the EVs. We are not looking for single biomarkers. We are also looking for signatures containing out of 5, 10, or a dozen of microRNAs, which gives you a much more specific information. And we want an early evaluation of cellular and physiological changes. And afterwards, these signatures, these microRNA signatures should be easily quantified via RT-qPCR. And we summarize more or less our workflow here in this slide and also in this review, which we published last year in Trillium Extracellular Vesicles. So the strategy is to test model organisms. We are working not only with humans, we are also working with cattle, and we have different target molecules, as I mentioned before. MicroRNA is in the focus, but as well mRNA or long non-coding RNA. Then we are using screening methods. In the beginning, we used microarrays. This is long, long ago. Nowadays, we are using RNA-seq, next generation sequencing. And to validate these samples, we are using, of course, RT-qPCR. And afterwards, we are using biostatistical methods to detect, to identify a biomarker signature. We start there always in a so-called training cohort, in a training set. And after we define the biomarker signature, we go into a validation cohort. This is a second independent patient group where we do just the same, but we are only using the biomarker signature and applying RT-qPCR to confirm it. And if we could confirm it, we can run the whole assay in additional cohorts, or hopefully in the future in routine clinical samples. So which disease we are analyzing or which we are interested in, we have a very good and long-lasting collaboration with the LMU, the Ludwig-Maximilian-Universität in Munich, and with the intensive care unit, the ICU, and therefore they have a lot of pneumonia and pneumonia-related sepsis cases, and also kind of variations of this disease, different stages. And of course, pneumonia and sepsis is under the top 10 global cause diseases with about 3 million deaths. And pneumonia and sepsis will lead to coughing, fevers, sweating, and so on. And our goal is to find for asthmatic symptoms and for pneumonia and sepsis in high-risk groups. And we want to detect novel and early discriminating diagnostic markers, which are highly required. And also we want to research the role of EVs in pathogenesis of pneumonia and sepsis. A few words to sepsis. Sepsis after infection comes to a high degree. First, you got a systemic inflammatory response syndrome called SIRS, where you have pro-inflammatory mediators. And with the increasing severity of sepsis, you're coming to a CARS, to a compensatory anti-inflammatory response syndrome, which then leads in high cases to organ dysfunction and hence to death. Therefore, sepsis is a very severe disease, and at the time of detection here, indicated at the very beginning, you have a survival rate of 85%, and with the time of non-detection, the survival rate really drops. And if you give very effective antibody therapy, then the survival rate starts to increase again. Therefore, there's a real need for speed in diagnosis, and therefore we want to help to diagnose sepsis and pneumonia related sepsis very fast and with new technologies and new biomarkers. So, but if we start a study, we have the cell, and within the cell we have a multitude of cellular bodies and vesicles, and the first question we have to ask ourselves is, which blood compartment is the best for microRNA biomarkers? Is this the full blood or all RNA, which I have in a liquid biopsy? Are they only the blood cells, or are these containing EVs? And even further then, which type of vesicle is the best? Are these the exosomes, the microvesicles, exosomes, or dropping down, are they all EVs? So this was the question for our first study. Therefore, we involved 14 patients, draw blood, and analyzed the exosome EV-associated RNA, the serum RNA, and the whole blood cellular RNA, all from 1 milliliter, all from the same patients. Our goal is, of course, biomarker discovery, and we are using next generation sequencing to screen it. And hence, we got the following results from absolute and relative mapping statistics of the small RNAs. And you clearly see that the full blood gives the best and the most reads in the statistic, but relatively seen, if you zoom it, you have in the exosome also a kind of concentrated and isolated information, which is similar, good, it gives a lot of biomarker candidates compared to the whole blood packaging information. The serum is a little bit lower. And the next question was how these biomarkers look like, and if we are looking for the differential expressed genes in these trial study, you clearly see that you have different biomarkers. Here it's depicted a significant different expressed microRNAs between sepsis and healthy patients. And you clearly see that you have in the cells an own biomarker signature, which is quite different from the exosome and serum family. They share a lot of common biomarkers. Of course, exosomes are part of the serum, and hence they share a lot of these biomarkers. And therefore, we concentrated in the future studies on the exosomes because we are thinking the exosomes gives us a very specific information. They are highly defined, we can isolate them, and hence we built up our biomarker strategy on these RNA related to these EVs. I just picked some of the biomarkers from this preliminary study. Of course, these are preliminary results, but we just picked some of these microRNAs, and you clearly see that some of these microRNAs could indicate and could discriminate between volunteers, septic and septic shock. Sepsis and septic shock, of course, two stages of this disease. Here we have 3 biomarkers, and if we put the patients or in different groups like volunteers, survivors, or non-survivors, even here we could detect early on first day of at ICU, we could detect possible candidates which may not survive, and this speaks for high severity and quality of the biomarkers. Of course, the outcome of this study was that we should use EVs for future studies. On the next level, we concentrated on the establishment of a so-called MySafe compliant workflow for the optimization of EV isolation methods from serum. The goal was to be fast and reproducible, reliable in the so-called daily clinical practice, and should be highly suitable for microRNA-based biomarker discovery. Hence, we included 4 commercial kits in these studies. We compared as well in this methodical trial 10 sepsis patients and 10 healthy volunteers. This was the mercury kit from Exicon, which is isolation via precipitation, the exospin from cell guidance systems, this size exclusion chromatography, the QEV from ISON. It's also a size exclusion, and also the chitin methodology, the ExoRNA, which is membrane affinity based. And we compared these 4, let's call it fast extraction methods with our in-house method. This is for us, it is and was the gold standard, is the ultracentrifugation. And the outcome of this trial was published 2018 in JEF, headed by Dominic Buschmann, my former PhD. Just to come to a few, just an overview of this paper. First, we've done a EV characterization. Of course, we've done surface markers via Western blot. We've done size and amount measurement via NTA, and we've done morphology of the EVs, all in the same amount of blood, 1 milliliter, from the different isolation types and purification methods. But the real goal was, of course, to perform a kit comparison with which kit system gives the best output in terms of microRNA signature. Hence, we took 1 milliliter aliquots from healthy volunteers, compared them with patients directly coming to the ICU, isolated them always 1 milliliter aliquots with the different methods, performed reverse transcription, performed RNA sequencing, small RNA sequencing, and at the end we made a differential gene expression and evaluated differently expressed genes, and then put these candidate biomarkers from the sequencing into statistical multivariate methods to get the best discriminating biomarker signature out. But first I will come to the characterization, and as you know, different isolation methods end up with different EV, let's call it subfractions or subfamilies. You clearly see here the mercury isolation gives us the lowest amount, the lowest diameter in EV, so the smallest EVs, but yeah, the range is quite high. And on the other hand as well, QEV gives very short, and exospin gives very short EVs. On the other hand, ultracentrifugation and ExoRNA gives or resulted in a little bit bigger EV. So here we have the averages of around 100 nanometer, and in the QEV, the so-called particles, let's name them particles and not EVs, are a little bit bigger in the range of 150 to 250 nanometers. So first outcome, different isolation from one and the same patient groups performed by one and the same person results in different subfamilies and subfractions of EVs. The EVs look under the transmission electron microscope. They look good. They have a clear membrane. This is also MySafe compliant to depict one, and or to depict a group of EVs, and hence to fulfill MySafe compliancy, we put these evaluation as well in the paper. Next part was the specific surface marker. Therefore, we of course had positive markers like CD63, 81, syntenine, or TST101. We had negative markers we have to include like argonaut-2 or calnexin, and of course, we included a protein contamination, which you see here down there. And the different groups came up with different Western blot signals. Here, the QEV gave, yeah, let's say the clearest, the purest result in terms of a protein contamination. Here we have syntenine, we have here CD63. On the other hand, we have samples with high contamination and with lacking information here on EV surface marker like CD63 syntenine, CD81 or TSG101. And then we questioned ourselves why this could be. And then we further on concentrated these samples via OptiPreg method in a density gradient with a density of 1.02 to 1.3. And then after, let's say, in fraction 8 and 9, we got an accumulation of these EVs, and successfully we could also find the right biomarkers in this subfraction and could confirm that we isolated as well EVs with mercury and ultracentrifugation. The next level was then sequencing. Later on, I present some more sequencing results, but here I want to point out that we also did a kit ranking for impurities in small RNA sequencing. What we mean by impurities, impurities are the amount of short RNA fragments in comparison to microRNA, are ribosomal impurities, due to impure isolation, and as well tRNA. And we included here a kind of lightning system, means green means this is very good, therefore we have low impurity levels, and red means you have high impurity levels. And here you see, clearly see the isolation before from QEV, where we got very good kind of EV-like particles, had high impurities in terms of short ribosomal or tRNA, and the mercury method more or less gives all green signals in all the purification groups. Here we have the volunteers and the sepsis, so we have 6 here groups which were compared. So we went more, of course, for the microRNA for our later studies. If you compare all the results from the biomarkers here in this group and put it here on the hierarchical clustering, we could see that we have 2 main clusters. One is containing from exospin and QEV isolation, and the second main cluster is from mercury isolation, differential ultracentrifugation, and ExoRNAs. And if you look exactly, we could see that even the methods subcluster, even ExoRNAs and as well as the mercury and ultracentrifugation. So this was the proof that there is a significant impact of the EV isolation method on the microRNA signature. Okay, so if you compare different papers, isolated the EV in different ways, you cannot compare the biomarker signature. You should always stay to your isolation method. Isolation is, in my perspective, the key issue in our microRNA development. Okay, after this hierarchical clustering, we went for the significantly regulated microRNAs, and of course, we had the highest candidates in microRNA, the highest read counts. Hence, we had the most microRNA candidates, differentially expressed microRNA candidates, here depicted by 144 candidates, which are exclusively in the microRNA group, significantly regulated between sepsis and healthy patients, followed by ExoRNAs, Exospin, and QEV. Only 19 microRNA were detected in all isolation methods. But of course, here we have exclusive candidates, which we can use later on for better discrimination of the patient groups. As I mentioned before, our strategy is also to validate the results. So we took here the 10 most highly expressed and lowest expressed candidates and validated these 20 microRNAs later on as well in RT-qPCR. And of course, you always will result in slightly different results, but at least the direction and also the strength of fold expression is comparable, which is depicted here in this correlation, where the correlation coefficient is 0.9478. So we could conclude that we could validate our biomarkers found in the sequencing. We could validate them independently in a single RT-qPCR microRNA assay. So concluding all these results, here we have a PCA of the most significant differentially expressed microRNAs. And if we are looking here on the mercury and on the ExoRNAs results, which seemed for us the best, we could clearly discriminate the healthy from the diseased, meaning clearly discrimination means in the PCA, the two clouds are totally separated. So this could be done with ExoRNAs and mercury. The other two isolations from QEV and exospin are also separated. So the centers of these groups are significantly different, but in some kinds they overlap. So for us, we continued with the microRNA isolation for the real patient study. So this brings us to the next question. Should we use arterial or venous blood for our studies? And all people are going for venous blood, but we ask ourselves, is there a difference between arterial and venous blood serum EVs? And for this study, we've done two purification methods because we started with a very, let's say, easy precipitation method. This easy precipitation method ends with high yield and low purity. We've seen these on this Western blot. And this time we also included a size exclusion. So we did one group with just precipitation, and with a second purification step, we additionally performed a size exclusion to come to a higher purity and come a little bit closer to a so-called EV after isolation. So we performed 20 patients. Here we have arterial and venous blood, and you clearly see if we compare the total reads after sequencing, there is no difference, and also the microRNA candidates, the read count of the candidates, there is no difference. Independent if we perform a mercury isolation and an additional QEV. But what we see here, we had slightly different microRNA candidate reads. So if we look here, the mapping percentage before with a crude precipitation method, we had around about 40–45% mapping of the microRNAs in the so-called crude EVs. And in the isolated EVs, we had much less, with around 10% microRNAs. But the question was, are these microRNA different? These are just the total reads. So we performed or compared it in a regression. That means here we correlated the arterial versus the venous expression from one and the same candidate. For the high expressed, this is A, and for the low expressed, this is B, up to an expression of 10,000 reads. And you clearly see, independent of arterial and venous, they behave similar. So the correlation coefficient is 9.99 for these so-called crude EVs. For the additional purified by a size exclusion, it behaves similarly. So this means there is no difference between arterial and venous blood sampling if you're looking for EV associated microRNAs. So then we performed a further analysis. We looked what is the variability within the patients. This means intra-patient, here black depicted, versus inter-patients from 2 times 20 or 2 times 14 patients. And you clearly see that the differences between arterial and venous within one patient is the lowest. So the difference between different microRNA candidates from healthy patients is higher in a different patient. So intra-patient variability is the lowest, and here inter-patient variability, either arterial or venous, is much, much higher. This comes to the conclusion that there is no difference either in crude or in additional purified size exclusion EVs, and here as depicted as PCA, and as well depicted as, sorry, my telephone is ringing and I put it down, as well as here in hierarchical clustering. And if you look exactly, you could even see here, go for B, that the patient samples like 15, 22, and so on group at the highest level. This means there is no significantly different expression between arterial and venous blood. Okay, with this first 3 papers, we went further on for real patient samples. Now we had the optimal sampling. We are sure that EVs are working. And therefore, we went for the first cohort. This was a learning cohort. And therefore, we started with 5 different patient groups. On the one hand, healthy, mild pneumonia, severe pneumonia, septic shock, and ARDS. And we compared these patient groups. These were 67 different patients. We performed EV isolation as shown. We performed RNA extraction, also via a QIAGEN kit. We performed small library preparation via NEB. Then we did a small RNA sequencing. And then we performed the multivariate data analysis, first differential gene expression and principal component analysis, and then PLSDA in different ways. In between, we did always our quality control steps. First for the EVs, so we looked at how the EVs are looking like, what's the amount, what is the size, surface markers, then of course RNA quality control, what is the amount, what is the RIN value, etc. pp. After library preparation, we had gel electrophoresis and so on. So we checked for quality of the library prep. And after our small RNA sequencing, there are also parameters which show you that the sequencing run works fine. And then we did a multivariate analysis, and even cis candidates were confirmed via RT-qPCR validation. And then after this training set, it turned out that we came to candidate microRNAs from this training cohort. And this training set also will be further confirmed in a second patient cohort with 75 patients to confirm these candidates via RT-qPCR. I will show now the results. Here, some small RNA sequencing mapping. Here you see we have volunteers, we have community-acquired pneumonia, we have sepsis, and different patient groups. Here are just 3 depicted from 3 patient groups. Look very similar. We had a strong select criteria or inclusion criteria. So every read count must be at least 1 million total reads per sample and at least 15% microRNA to be included in our further study. Here are some significantly regulated microRNAs shown, which are always compared to healthy volunteers, and from different pneumonia types like mild and severe pneumonia over to ARDS, acute respiratory stress syndrome, up to septic shock or sepsis. We had 19 overlapping microRNAs, but we also had some discriminating microRNAs, which are specific for different severity and which could, of course, possibly distinguish between mild and severe pneumonia up to the severe cases of ARDS or sepsis. But if you group these kind of candidate microRNAs, this was one of our first trials we've done here in the PCA. You could clearly see that the volunteers group very good together, or group in the lower right corner, but all the other patients are not grouping in any patients group. So this was the first, very, not very, but a little disappointing result from our first principal component studies based on differentially expressed microRNAs we found. And then we implemented supervised clustering, the path lean square discriminating analysis, or short PLSDA. Where is the difference? So for PCA, you test for groups, you want to discover pattern trends or even detect outliers like this guy out there. But with the PLSDA, you tell the software what are your groups, what are the group members, and the software is looking for the best classifiers separating these groups. So outcome of the PLSDA is a classification and are potential biomarkers from multiple treatments. You can use 2, you can use multiple up to 5 or 10 treatments, and we perform these PLSDA in our perspectives. And here, okay, this should rotate, but it's not working. To see a little bit better how these clouds behave. So I will stick here to the non-rotated version. You see the different patient class here. So the volunteer is a very, let's say a small and very concentrated cloud. The severe pneumonia is kind of in the background, is far behind. And then the mild pneumonia, the septic shock, are more or less in between. And if you group these here on a, if you plot these here on a ROC curve, you can clearly see based on 11 candidate microRNAs, which turned out to separate the groups in some manner. We could discriminate the volunteers, of course, from the others. Very good. This is this pink ROC curve, but the other patient groups could not be separated in a very good manner, like here's a septic shock from the others. And this would be very important to have the septic shock separated. And even more important is to find these intermediate stages like mild pneumonia separated from the other patients group at the first day at ICU. And then we handled or imported these found candidates into pathway analysis to see, does these microRNA make sense? Are the candidates really relevant in a physiological manner in sepsis? And hence we came out, and I just depicted here a bacterial derivation. So 39% of our genes in this pathway are regulated by the found microRNAs. So it seems that we found the right microRNAs. But coming back to that, we could not separate the 5 patient groups. That's why we focused on 3 patient groups. Of course, the volunteers, the healthy is one group. The community acquired pneumonia is the other group, meaning the mild disease group, and the severe groups like the sepsis, like the RDRS, like the severe pneumonia. These were summarized in one group. And this, again, it's not rotating. I'm sorry about that. It should be animated. With these 3 groups, meaning the healthy, the mild, and the severe patient groups, we had a very good separation. We had 12 microRNAs in these biomarker signature, and these 12 microRNAs could separate the groups. And if we afterwards we confirmed these candidates in RT-qPCR. Yeah, here it is. We confirmed these. The correlation coefficient are not that good like they were before, but at least we had similar regulation pattern in PCR, in the training cohort, and also later on in an independent validation cohort. Here also the results of the validation cohort are implemented. So it seems that it worked and that these microRNA signature is working. And here you see the result of the validity of these 12 microRNA biomarkers in these 67 patients. So from this training set, we had an accuracy of 92 to 99%. So of course we could separate the volunteers from the others, from the sepsis and the mild pneumonia with 99.2%. And we also could separate based on these biomarker signatures, the sepsis with 99.6%, and even the mild pneumonia could be separated from sepsis and from the volunteer with 92%. But this was a training cohort. And then we performed the same analysis in the validation cohort. So that means only these 11 microRNA were, sorry, only these 12 microRNA were used in these 3 patient groups in a totally new patient, independent patient. And we had a prediction accuracy based on these biomarkers for community-acquired pneumonia of 83% and of sepsis for 78%. As an average, we gained for 73%. So it looked good that these biomarker signature could predict the stages of sepsis or the severity of sepsis at the first day at the intensive care unit. And this was the outcome of the paper which we published last year. So in the middle of these biomarker signature are 3 microRNAs. They are markers for community-acquired pneumonia. This is 193A, microRNA 542. Okay, we have to delete this double microRNA, and the microRNA 1246. And as well, here it's depicted just as a single assay, RT-qPCR assay, and you could clearly distinguish between volunteer subs and CAP, volunteer and CAP, and for these 1246 for all groups. And if you use these 3 as a kind of mean value, you could clearly separate 3 patient groups. So for us, 193A, 542, 3P, and 1246 are marker for CAP, and 1246 is a marker for sepsis. This is embedded in a so-called canonical network. So you see here 193 and 542, 3P. These are the, let's say, the centers of this network. And if you ask ingenuity, pathway analysis, IPA, it comes out with cellular and humoral immune response, antimicrobial response network, and pathogen influence sequencing. So this all goes in the direction of pneumonia and sepsis. So, and let's come to the end of my talk. Last year and this year we have COVID-19, and also COVID-19 is lung associated, is pneumonia associated. Can we translate these results as well to COVID-19? Hence, we started further collaboration. We started first samplings. So we had several studies. I just want to introduce you to one of these studies. So first study is differences of COVID-19 community-acquired pneumonia and RDS compared to classical pneumonia and classic sepsis patient. I will not go into detail of this study. For now, we have 60 patients in this patient group. I just want to show you how the COVID-19 biomarker pattern is changing over the days at the ICU in 15 patients. And in future, we also include in this study so-called cured convalescent patients. We just started to vaccinate people and sample them compared to the real disease patient and want to compare them. And of course, we have a cohort who's continuously healthy. I just want to focus now as a first result on the second group, where we followed patients during the ICU from day 0, 4, 7, and 14. Again, some quality controls, EV sizes, EV concentrations from different patients. So the particle sizes are in the range of 10 to 11, 10 to 12 particles per serum. We are talking about a real EV, which we could isolate, and the size is quite normal between 100 and 150 nanometers. What I want to show you, we found different expressed microRNAs. So see, I have to say these are all preliminary results on 15 patients, but we could see differences over the day of COVID-19 at the ICU. So we have here day 0, day 4, day 7, and day 14. And of course, we have a trend of varying microRNA pattern. So there is a progression of the disease during the stay at the ICU. And which microRNA are involved? We have here, of course, our candidates, which we've seen before, 543. We have new candidates, and we also have new influenced networks. I just depicted this green one. This is an olfactory pathway. This is an olfactory receptor. And we all know that smelling is not that good anymore after a COVID-19 infection. And here we had a clear regulation of found microRNA in our signature on these olfactory receptors. This is just as to depict that there is a first result on the olfactory pathway. So as a summary, we established a massive compliant EV characterization and fast EV isolation workflow. EVs show more significant results compared to serum or cells from whole blood sampling. There is no difference between arterial and venous sampling. The mercury EV isolation showed the best candidates for biomarker signature. For the development of candidate biomarker signatures, a training set with 67 patients was performed. We could find candidates which we could confirm via RT-qPCR, and these markers could predict in the validation set the community-acquired pneumonia and sepsis between 78 and 83%. And for the COVID-19 biomarkers, we have first preliminary results. There are more to come. We are still sampling and we're still analyzing, we're still sequencing, but we see changing differences between day 0 and day 14, and also different severities and patient groups compared to classical IVS and community-acquired pneumonia versus COVID-19 patients. The outlook, we have specific subset for the early diagnosis of community-acquired pneumonia, the progression of pneumonia, and also for COVID-19 cases. We have, on the one hand, commonly expressed signatures with our microRNAs 542, 1246, and 193A. 1246 may be also an increased risk marker for sepsis. We also found these microRNAs in COVID-19 patients. We find also of clinical importance as identifying the normal cup at first day at ACU and separating the high-risk group for like sepsis or RDS, and identifying for pulmonary infection based on EVs. MicroRNA has high potential as treatment and for early intervention. Therefore, a secondary complication such as sepsis could be prevented. For the outlook, for the successful use and of course for biological validation of these microRNAs, we need additional and larger studies and larger patient cohorts with multiple conditions, and these samplings are ongoing. And as acknowledgement, I want to thank, of course, our group here, Dominik, Benedikt, Steffi, and also Christine Wurmser from the core facility. They're from 2 clinical hospitals in Munich, Agnes Meinert, Anja, Melanie, Florian, and Gustav and Marlene and Otto Steinlein, from a company which was involved in identifying the biomarker signature was Stefan Koschotke and Michael Bonin. And of course, we are financiers, where we had Bundesministerium für Wirtschaft und Energie, zentrales Innovationsprogramm für den Mittelstand. And for the COVID-19 project, we had the Bayerische Forschungsstiftung and recently the TUM Universitätsstiftung. And at the end, I want to say thank you to everybody. And here, these are pictures from better times where we had fun at the Toronto meeting or at the GSIF meeting in Germany. And 2 years, it's already 2 years ago where we met in Kyoto with our groups and other collaborators here. And I thank you for your attention. And of course, I'm open for questions. Thank you very much.
Michael, it's a wonderful presentation, and
thank you.
Successful strategy in discovering microRNA biomarker. that is associated with EV. So we have already quite a few questions here. So first is Wolf Holthorner. Please go ahead.
Okay, thank you very much, Michi, for this nice presentation. Hi, hi from Vienna. Maybe I didn't get it, but if you isolate the EVs from whole blood, do you use serum or plasma as the starting material?
Here we are focused on serum. This was mainly due to the sampling strategy at the hospital, yeah.
Okay, yeah, because we are also planning now to work with EVs, and I think it's not that easy if you use serum or plasma, and I think you might end up in different EV populations. Do you have any idea about this, or?
I only have the knowledge of the papers, but we did not investigate it. We did the arterial venous comparison, but not plasma serum, sorry. But this is always due to the ICU you're working with. And normally we are quite happy to get a lot of patient materials, but ICU is always a problem because the patients are really stressed and under severe condition. And even under COVID-19, we have, let's say like this, we have problems for good sampling. Of course, the people are, especially in COVID-19 sampling, are under high stress. And we clearly see the results in lower sample quality and lower read counts, okay? So sampling is also very important. I want to mention this here. Yeah.
Okay, thank you very much.
But no idea and no recommendation of plasma very serum. But there are several studies out there. Thank you.
All right. The next question is from Ahmed Gat, but I will read it because he doesn't have an active mic. Okay. Which cutoff criteria or threshold do you use or recommend to consider a microRNA as an express microRNA in the sample?
So thank you for the question. We have inclusion criteria as from the read count. So we are just going for the good read counts. I mentioned this, at least 1 million reads, okay? And then for microRNAs, and then for the exclusion criteria for what is a good microRNA is classically is a twofold regulation. So a candidate with a twofold regulation is kind of good candidate. Oh, I know. It is like it is. It was always that way on mRNA expression, different expression, but this will give us the first candidates. The first candidates will give a twofold regulation. And in terms of expressing high, we started with at least 50 read counts as an average in all samples. Of course, if you have a biomarker, the biomarker should be present in all samples, the healthy, the diseased, and in all patient group to make a comparison. If the biomarker is absent, okay, it's maybe the optimal biomarker because it's absent in one group and it's expressed there, but then you can never do a differential gene expression because then the differences are too big, okay? So it should be at least 50 reads in all the samples, and it should appear, it should be an existing expressed biomarker in all samples. Thank you.
Okay. Toluela say Awoyemi, please ask your question.
I can read the question, but actually I cannot really answer it.
I'll read it for you, Michael.
Sorry,
so,
maybe I can.
What anticoagulant?
I read the question, but I cannot answer it. I'm really sorry. Maybe, I think I've seen Steffi. Steffi, are you here? Do you know it? Steffi Hermann? Or Dominic, is Dominic here? Or Froni maybe could also know it. But we've done never studies on different coagulants, as far as I know.
Can you hear me?
Yeah, we can hear.
Yeah.
Okay, so usually it's EDTA, which is used because citrate can hamper the downstream analysis in in a lot of ways, which is not that, yeah, extensive when you use EDTA samples. This is maybe the reason why this is the sample type which is used more often. And also, if you want to validate your results with RT-qPCR, then you should also go for EDTA samples. I think, yeah, there's a lot of overlap in techniques that prefer EDTA samples in that way. But yeah, if you, the choice what to use, so serum or plasma, it really depends on what samples you get from the clinics in that case.
I think this is the most.
It's really availability then. So how we're concerned about platelet microRNA contamination in the serum? Is it possible there's a contamination of from platelet microRNA?
We never analyzed this, but yeah, no, cannot answer on that. Sorry.
Sorry, my question about the citrate and EDTA. The reason why I'm asking is because I know one of the recommendations by the International Society of Hematology says.
I can't understand it.
Can
you help me? I cannot really understand it.
Yeah, it's your connection is a little bit off, so yeah. It's just really hard.
He asked the same question we already answered.
Yeah, he's the person who asked
That's what I picked up.
Yeah.
And the platelet contamination, yeah, I cannot comment on this. Maybe somewhere else in the auditorium can answer this, but we never purified platelets. Yeah, you have to set up a study, but otherwise you cannot solve this problem.
Yeah. Okay, that's okay. Let's go to the next question from Sabine Bartel. Please go ahead.
Yes, thank you for the great talk, first of all. I was intrigued by your COVID data. So I was wondering if the microRNAs change over time in the ICU. This could, of course, also be because the patients are treated. But I was wondering if they were treated the same way, or if you maybe even found a microRNA that could indicate that some medication would work in one patient, but not in the other one, sort of a treatment biomarker.
Good question, no answers that far. The problem is we are just started. We just started the sequencing. These are the first results. You know, you're always happy to see first results, that there are differences. As I said before, we got money from the Bayerische Forschungsstiftung, from the TUM-Universitätsstiftung. We enlarged our cohorts as shown, and treatment, medication, all this stuff is not present in our hands now. So we had to analyze it later on. For now, you're fully right. You're fully right. We cannot say it. Is it the medication? Is it the treatment? Is it whatever? I don't know. I don't know. I really can't say it so far. Okay. We are just at the beginning, and we try. We have different cohorts, and you always know you have to combine these cohorts. And we have reconvalescence patients, we have healthy patients, age-matched. We have blood samplings over time after a COVID-19 infection, and the same after infection to see, after vaccination, to see the differences between a real infection and an artificial vaccination. And then out of this, we want to learn. But this is much too early, much too early. We're still sampling, and yeah, maybe next year, same time. Thank you.
Okay, so next one, and I think Isabel Diaz-Lozano has two questions. Isabel?
Yes.
Hello.
Hi, very nice talk. Thank you so much. Very nice also overview around all the methods that you have used. So my question is about when you isolate ABs by size exclusion chromatography, I have seen in your workflow that you concentrate with Amicon. Have you tested if there is any difference when you concentrate the ABs with Amicon and the microRNA profiling that you get from them if you don't concentrate it?
Good question. And we concentrated just in one isolation. And I also should hand over this to Dominic. Is Dominic here, Vroni? You know it? Because he did the work.
No, he can't join, unfortunately.
He can't join, no. The concentration of Amicon was done, yes. And it ended up, of course, with different slight results, but I cannot answer this in detail. Sorry for that.
No, it's fine. And then my other question was about...
You can contact Dominic Buschmann. He done the study and he knows exactly which to which content he concentrated and so on. But anyways, we could not see the differences because we didn't have the results from Amicon concentrated and non-Amicon concentrated. So we could not see the effect of concentrations. The concentration had these that we want to trace back to 1 milliliter of patient's serum, okay? Because we want to compare the analysis on 1 milliliter. Therefore, we had to concentrate to come to more EVs, which are then leads back to one, or EVs, which are more or less isolated out of 1 milliliter, yeah.
Okay.
That was and also due to the RNA concentration, yeah.
Yeah, and then my other question was about the hemolysis. If you have seen any differences in serum samples that they have hemolysis, and then the microRNA profiling. In those, you haven't had any, or you haven't considered the hemolysis in the serum samples in terms of the microRNA.
Yeah.
Yes.
Since I did not perform the samples, I cannot tell you about the hemolysis, but maybe I can also trace back to Vroni in another study. He done a lot of work with our blood doping study. Vroni, do we have any hemolytic samples in our blood sampling study? And could we find any differences, or do you know something about that?
Yeah, so I got my samples from the clinics and they checked it for hemolysis. So they checked for free hemoglobin, but you can also check for it if you compare the ratio of two specific microRNAs, one of which is the microRNA 451, and the other one I have to look it up. But if you set those two microRNAs in ratio, you can estimate the hemolysis rate.
Okay, that's great. I didn't know about that. Thank you.
You're welcome.
Yeah. Great.
Thank you.
Next question, Maimona Almasawa.
Would you consider studying other types of small non-coding RNA as partial biomarkers? Yes. Good question. Very good question. Let's start. We are coming from the mRNA field. So I did mRNA analysis since 90s, early 90s, where we started with first PCR. And then we came over mRNA, microRNA. And then we had a time where we had these nice peaks at 30 plus of the small RNA sequencing peaks, and we thought these were piRNAs. So we've done a lot of studies. This was mainly done by Benedikt Kirchner. He's not here, I think. But it turned out that this is not piRNAs. These were the new candidates about 5 years ago, piRNAs. And it turned out these are tRNA halves, okay? So often you get a microRNA peak and then a second peak, which is often the same high, as this is in our sampling and isolation method, a tRNA halves, not piRNA. And of course, some people using piRNA, others are using as well tRNA as candidates today. And as I said before, we are starting now, we've done mRNA analysis as well in EVs and with long non-coding RNAs. And to be honest, so far, the long non-coding RNA are not that successful as we expected. we found no biomarkers, but this could be due to the bioinformatical tools, I have to say. Long non-coding RNAs is very diverse. It's very diverse. And hence the bioinformatical analysis is not that good and not that stringent, okay? So we are still struggling with long non-coding RNAs. And then we still have one family which is always very interesting, and a lot of rumors are going around. These are the isomers. And also Benedict programmed a whole pipeline. This was around 2-3 years ago, and we have a full working isomer pipeline. And we thought maybe the isomers are the key. So we are just always talking about the canonical microRNAs, fine. But even the isomers which are laying behind the canonical with variations, mainly at the 3' end, there are lacking, there are missing, there are additional polyadenylations mainly, is the key. And there are a lot of publications out where say cancer tissue, where you have highly active tissues, highly diseased, they may change their microRNA profile and produce more, let's say, isomers in a wrong manner because the enzyme do mistakes. For now, also very disappointing in our perspective, we didn't find any studies where we analyze, where we really find, let's say, different microRNA pattern, which are significantly different from the between healthy and diseased, which are based on differentially expressed isomers. So,
how about Small-RNA, Michael?
How about
the Small-RNA?
Yeah,
I mean, are they potentially useful?
So one example would there be a very good candidate to ask for. He's done a lot of analysis of microRNA. We are focusing on microRNA and isoforms, but yeah, others do tRNA, others do piRNA, long non-coding RNA. Yeah, but we are focusing on the microRNA. And next important thing, you can use it as a predictor, but afterwards in the clinic, you need a simple, stable to test to perform your signature in the clinics. And hence, these isomers are only differing in one single base. And therefore, the diagnostics later on in the so-called routine assay, in the validation, in the clinical routine, is very difficult, okay? Therefore, just canonical RNAs, which gives you more validity in the later on assay, I would say, yeah. Thank you for the question.
Yeah, okay. Last question is from Tahara. It's also my questions actually. How do you normalize it?
Normalize what?
Hi, Toshi. Toshi is here.
Hi, Toshi. How are you?
I was wondering if it's you.
Oh, okay. Oh,
okay,
sorry. Okay.
Yeah.
So, so the best major problem is you always normalize the microRNA, So everybody normalize a different method. Yeah. Yeah. So do you have any idea to optimize the normalization?
Yeah. So the sequencing before the whole method is kind of standardized, but the sequencing will be normalized about the classical DSEQ packages. And later on in the PCR, we are using normalizing microRNAs. This is Small-RNA, where I put it somewhere. Which we use for normalization. Where I have it. It's UniSP6 and microRNA 30D. Okay, this we are using for normalization, but I have to add on, we are working on a paper. We are working on a paper which gives the most, which we submitted, and we're waiting for review comments, where we test for good normalizer, because normalizing in EV isolates or on single microRNAs is a problem, I know. And that's why we established an algorithm called myref. and I presented this on the meeting, on the ISEF meeting in Ghent. It was December 19. And this is now for application. And therefore, we have a software where you can upload all your microRNA. Of course, we first focused on blood-derived microRNAs, EV-associated blood-derived microRNAs, and they will come out with this most stable. And there is 30P and, of course, others candidates within. But hopefully the paper will come out, yeah, in 1 or 2 months.
Oh,
and then also we're collecting data from other blood EV-related plasma serum, full blood, and then, but more or less it's the same like we do it before. It's like a genome, best keeper, and so on, algorithms in the background, looking for the most stable reference gene, reference microRNA EV associated. Yes.
Okay, thank you.
Welcome.
Okay, now we're actually over like 14 minutes, but there's another last question. Would you like to take that, Michael?
Yeah, of course.
Okay, okay. So Zoe, are you?
Hi there, Michael.
Okay.
Yeah, thanks for the nice talk. Totally worth it. So I have a question, like, accessing or recruiting precious patient samples is not always an easy hit. I saw that you started working, or you're working with 1 ml of respective sample. How would you comment on the minimum amount of sample needed for microRNA profiling?
So, good question. From my perspective, I can't give you any recommendation. I know from Joe Wangesampel's group, they're doing microRNA profiling in, as far as I know, 300 microliter blood.
Okay.
That's what I know. But maybe anybody else can comment on this. Possibly, if the quality is good, if the sampling is good, and EV isolation is good, and everything is validated, I'm pretty sure you can go even lower of that, okay?
Right.
But from our group, we cannot have any recommendation. We are working with clinicians. We always get 1 vial, and then normally we have it in access, to tell it like this.
Yeah.
If you are from biobank samples, maybe 200, maybe 300, for sure 0.5 ml, but lower than 200-300, I cannot comment on this. Please look in the literature and read papers of the Joe Wangesampel groups that are doing a lot of biomarker research, and from BioGazelle, old friends. I know these guys since 20 years from the RT-qPCR conferences. Say recommend at least 200 to 300 microliters.
Yeah.
But
Thank
maybe
you very much.
Anybody else can comment.
Hey, I think that's the end of our discussions. Thank you so much, Michael. It's an amazing talk. We learned so much about how we should set up and work through our microRNA candidates, and hopefully we managed to get a great result as you have shown today. Thanks so much for sharing your strategy and tips.